Welcome to New York Bio's Virtual Breakfast Series, a digital program started in 2020, bringing you fireside chats with leaders across the healthcare spectrum. This week's episode features Matthew Malone, Senior Vice President at Taconic Partners.
All right, good morning, everyone. Thank you for joining us again. My name is Jennifer Hawks-Bland, and welcome to our New York Bio Virtual Breakfast Series. We're excited to have Matt Malone from Taconic to talk to us this morning about all things real estate, development, life sciences, and maybe some other random things we don't know yet. So we'll get into that. A special thank you to our sponsor for this month's series of breakfast, Avantor and VWR. They are our partner for lab supplies and lab build-outs. So we really appreciate their engagement in our ecosystem. As always, if you have questions for us as we go through the conversation today, please put those either in the chat or the Q&A, and Derek and I will look out for those during the course of our conversation with Matt. Always also a privilege, point of privilege. New York Bio's annual meeting is coming up on September 27th. We hope that you will register and join us if you haven't already registered. I will in the chat for everyone put a discount code in case you missed out on the early bird registration. Just for joining us on our virtual breakfast, you qualify for that same early bird discount. So I want to give you that code and I'll put it in and you can use it to register at your convenience. With that, I will kick it over to Derek to kick off our conversation with Matt. Thanks so much.
All right. Well, good morning, Matt. It is wonderful to have you here. There is so much to talk about because you've really been at kind of the center of what I'll call the real estate evolution in New York City. I'm very, very excited to talk about this. And as you know, we usually start with an origin story for all of our guests. And it was a toss-up this week to figure out whether we would start with how you got into real estate and architecture in the first place, or whether we should start with the fact that you grew up one town away from me and we kind of semi-overlapped in high school. So I know which one I would talk about if this was, the virtual beer session, but I think we can probably go with the former one for this.
Yeah, well, it wasn't just, overlap in high school. It was rival high school. So let's call it.
That's true.
Oh,
okay.
No, we don't want to go there then.
Sorry, everybody. This hour is going to take a much different turn.
I'm going to have to do the whatever.
Jennifer's going to step in.
It's an interesting perspective nonetheless, because it does play into where we grew up, for lack of a better way to say it, does play into some of, where I find myself today. I say some of because it definitely took a left turn for me. We grew up in northern New Jersey. We grew up in an area, immediate vicinity to. I had neighbors and friends whose parents worked at Becton Dickinson because the headquarters was just down the road. Not too far away in another town called Wayne, New Jersey, was an American cyanamide outpost. So there was a lot of, as it's no big surprise, science and technology in and around our environment. But, in northern New Jersey, it was probably a little less prominent than it is in, say, central and southern New Jersey. If I was to kind of put a finger on that, I would say the two that I mentioned are probably the two really most prominent that existed in that area.
Yeah.
So when we were in high school, I was only peripherally aware of it. I wasn't necessarily a science guy. It wasn't truly in my psyche. I was a bit of a sports guy playing some soccer, for a pretty competitive team when we were in high school. And I enjoyed that, went to college and basically had made the decision in college that I wanted to focus on the arts. I was, a bit of an artist. I drew a lot. I did sculpture. I was very much into, AP art classes and such. And I went to Syracuse University because for me, it was an opportunity to get to see, big time sports, which I thought was fun. But also,
sorry, my SEC hat is.
See, Jennifer, I don't want to go here either. I'm just going to say. Back in my day, Syracuse was beating Auburn on a daily basis.
Bless your heart, Matt. Bless your heart.
I don't know what that means.
Watch your mouth, Jennifer.
But that being said, no, I think, the idea was for me, going to a school that was very reputable from an architecture perspective and really kind of getting, to be honest with you, a bit of a slap in the face. And it was a positive thing. I had always kind of assumed architecture was a little bit more on the engineering side of things. And I kind of felt like, okay, math and engineering, and that's what architecture is about. Let me make sure I go to a school that if I don't like it, I can transfer to the art program, right? And Syracuse had a pretty good art program. I fell in love with architecture within, the intro class, because they basically said, you've got it all wrong. This is as much art as it is math. And I just completely dove in. So fast forward to, being around an environment where, and I had two roommates, one of whom played football, the other played basketball. And ultimately kind of dove into this idea of the melding of architecture and sport, or architecture and entertainment, right? So I came out of school saying that's what I wanted to do. I wanted to be an entertainment architect, and interviewed with and got a job with a firm in Princeton, New Jersey, that had done a lot of work at the time for Six Flags. And I said, okay, well, this is an opportunity to kind of get my foot in the door with that. There's entertainment components to this. There's ride design. There is auditoriums for the shows that they do and things of that nature. And no sooner did I get there that they said, hey, guess what? Six Flags has decided that they're going to completely pull back all of their projects, and we're closing our entertainment division. I mean, I couldn't have been there for a month, and that's what came about. And they said, so we do a lot of work in laboratories, and we wanted to know if maybe you wanted to pivot yourself to something in laboratory architecture. And immediately upon that happening, I said, well, I want a job, so I'm going to stick through this. Yeah, absolutely. I just landed here. I don't want to necessarily go on the job hunt all over again. Got into the laboratory side of this stuff, working for big pharmaceuticals, Merck being kind of the first one that I really engaged with as a small projects team member. And so we did these smaller laboratory renovations for predominantly the big pharma that was up and down the Route 1 corridor, as a Princeton-based architecture firm would. And what it enlightened me to on that small project team was I was engaging with the scientists, and I was able to employ some of the things that I had learned about learning in architecture school directly, and then correlate that to a design output. And the other premise was that design output was helping these individuals create cures and create things that improved well-being, healthcare, society to some extent. And I felt like that was incredibly rewarding. And to be honest with you, the fact that I backed into it with that small project team was particularly helpful to me, because it exposed me to a lot more of the project breadth. So I was at the front end of these projects and all the way through to the back end of the projects, to the ribbon cutting, where some of my coworkers and colleagues were on much larger corporate campus type projects that were, focusing them. The big joke in architecture was, oh, when you get out of school, you're going to work on toilets and stairs. And that is what they were doing. That is what they were doing. They were, oh, I'm working on the big Pfizer project. And I said, you're doing the toilets and stairs.
Important components of the workflow of the day.
Yep.
Like I said, it helped me because I got to engage scientists. I got to ask the questions of them. And I did. I took advantage of it. I said, I know very little about science. Going back to high school, I did poorly in science, in my opinion anyway. And I think it was because of my interest in it at that point, just wasn't where it needed to be to truly take something from it. And so I came into it with this, I don't know anything about science. And all of a sudden I'm standing in front of a PhD. I better start asking questions and learning as much as I can. I have, a small period of time at which I can be the idiot. Turns out I can still be the idiot today. So let's just, ignore that fact. I ask questions as much as possible because it's the best way to understand from the individuals who need to use the space what they need, and offer to them some of the things that I've learned along the way. So I mentioned I started working in pharma. My project scales grew as I continued to learn more and more about it. My organization kept putting me in front of people. It ultimately resulted in, the pharmaceutical market in New Jersey and globally had kind of recoiled a little bit, right? And it was just around, 9/11, let's just say, that you started to see chemistry and pharma kind of recoil a little bit as to how they were going about it, because they were living on blockbuster drugs, and it was very, very difficult for them to continually find the next blockbuster. When 9/11 happened and everybody got very concerned about bio warfare and things of that nature, we saw a lot of federal work kick up, and I wound up moving to DC. Specifically, our office was in Bethesda and directly serving the NIH as a client, and doing a lot of work for biological laboratories, bioresearch. And one of the bigger projects that I was working on down there on the NIH campus, the Bill Young Building 33, which is an NIAID facility on the campus of NIH. And I got to learn more than I ever thought was possible about things like high containment, things like, BSL-3 and BSL-3 enhanced laboratories, BSL-4 for that matter, but didn't really get myself into too much BSL-4 work, and learned that there was another layer of expertise that existed above and beyond where I was at, and I was only just getting the surface of that. But again, same argument, really, really interesting learning process, and understanding not only the mechanisms that are the federal government and the way that they run projects, but also the individuals who are working there and what their goals are and what their aspirations are. And one recurring theme I'll bring up is that it really is, at the end of the day, a lot about the people that inhabit the space as an architect. And that is so important to me. And that's what, in my mind, makes more successful designs. So fast forward a little bit further, NIH decides they're going to put a lot of their funding into the academic world in order to get further research. And what happened was, as a result, because the NIH and the federal government has oftentimes said they didn't like the idea of being a landlord. It wasn't really what they were best suited to do. So a lot of the funding went toward academic, and as a result, so did I. I came back up to the New York area, and I was focused on academic architecture, academic science, doing work for a lot of the major institutions that were doing R&D. So I think along those lines, I got involved with some work, at Rutgers University, Princeton University, Yale University, and some smaller universities in and around the areas as well, NYU, Columbia, etc. And you started to understand the difference in academic design, which was more focused on the individual researcher at the time, from, say, corporate design, corporate pharmaceutical design, which was focused on departments a little bit more. And I think the federal was actually a blending of the two of them. So with that armor, or that knowledge, if you will, it was an easy kind of morph into a little bit more of a corporate eye towards some of these things. And when I say corporate, I mean this idea of the burgeoning corporate or commercial life science market. There was a healthcare stop in between there where I did some nice translational work, and that was another thing that was fabulous to see, and I think still serves, because I think it is something that's very important today. But ultimately got to focus on the advent of commercial life science architecture, as it was kind of happening.
What is translational work from an architect's perspective?
Well, so it's interesting because it has, as with everything in science, it seems, it has so many facets to it. So when I said translational, what I was referring to specifically was this kind of, put it in air quotes, the bench to bedside mentality of bespoke healthcare. It also is tech transfer as well, when you talk about translational, going from a bench to a process. And I think both of those are valid. And I've done some chemical production facilities as much as I've done some healthcare work. But from that perspective, I think that the translational element is an important one as we kind of walk forward in this conversation.
And tell us about kind of the process, because I like the idea of drilling down a little bit on the sort of corporate versus academic and sort of how they think about, and clearly I get the department versus an individual sort of researcher, faculty member, but like, how do they approach it so that you literally have to design it differently? Because I kind of would have thought it all looked about the same. Maybe you had different types of people in there.
I think the major differences tended to be, and none of this is absolute black and white set in stone, but the major differences tended to be that in the kind of corporate pharmaceutical world, depending upon what department it was that you were dealing with. I mean, drug safety evaluation facilities, which I've done a handful of, are very, very specific and have, a little bit more of a regimented chain of custody, and a little bit more of a kind of a production line element to them. But in the research side of it, a lot of times you tended to see larger and more open laboratory environments in the corporate world, simply because you were dealing with kind of a corporate oversight, and it kind of, I don't want to say neutralized, but made everything a little bit more equal and easier to serve from their facilities group's perspectives. In the academic world, as with NIH, it was a little more grant-based and individual science funding. And so as a result, what you got was a little bit more of a compartmentalized approach to the laboratory environments. So you had smaller compartments of laboratories. And that's what kind of led to all these challenges of, well, how do we create collaboration if we're compartmentalizing everything? And I think that's a major kind of back and forth between the way corporate has done it and the way that academics have done it.
Mm-hmm.
So the corporate needed it to be more fungible in the long run, where
Yes.
if they change projects or whatever, they could without having to redesign and re-engineer an entire space.
Right.
They could move another project or another piece.
And I think the corporate, you got more involved, and I apologize, Jen, for cutting you off there. The corporate got more involved in things like shared equipment because it was all one company, right? Whereas if it's grant-based, every scientist has got their own equipment that's part of their grant. And in some cases, the grant doesn't allow that equipment to be shared. So it's a slightly like a different mindset in that regard as to how you kind of approach these things. And to be honest with you, and I hope I'm not offending too many scientists by saying it, but they tend to become pack rats. You know, they've got a laboratory full of stuff that they've had in that laboratory for 25 years. And you say, maybe you should think about getting rid of that. Or maybe that chemical you got in that cabinet over there is no longer, viable. You move on, get a new stock. And, I've had this for 25 years. This is how I do my science.
Might need it. Yeah.
And that's the scientific method. I want to change nothing. And that way I'll be able to see the differences more easily, and that in that understanding that is how I've been able to, throughout my career, been able to talk to the scientists about, well, we're changing this, but it doesn't really change that, or this is an improvement and you should see better results from your science as a result of it. It's not always an easy conversation, and some folks get set in their ways, but that's the reality of the situation.
I think that applies to lots of areas of life.
It does.
So when you got to New York, startup lab space in biotech, I imagine wasn't even a glint in someone's eye, let alone a thing that you would think had actual momentum.
I would say, just previous, like I said, when I was working at the NIH, was actually about the same time there was a considerable shift in the overall industry's approach to chemistry versus biology, right? So I'm in like 2002, 2003, somewhere in that vicinity when it was that, because I was working for the AID and because we were focused on BSL-3 and SARS, and believe it or not, it's funny, it's I go back on it, and COVID happens in 2020. I said, this reminds me a lot of the 2002 outbreak of SARS, and everybody goes, well, it's the same darn thing. Coronaviruses, and it's a whole thing, but bio kind of started. I don't want to say it started, because I'm going to short somebody from the credit that they deserve, but it really became more prominent right around that time. Biology became more prominent in that kind of 2002, 2003 area. And that's when I think you saw people saying, well, what do we do with bio? How do we do this? Because chemistry didn't necessarily support, because of the challenges associated with it from code and from building requirements, didn't really support a lot of startup, in my opinion. And the benefit of chemistry was it's so easily, replicable. But because I say easily, I've offended more people, but because chemistry is something more of a mathematical absolute, whereas biology is a living thing, and it becomes a little bit more difficult for you to replicate consistently. And I think the beginnings and failures, if you will, of some of these startups is based in large part on that. Like, we did it once, but I can't replicate it. It's really challenging. So when I came to, as you say, when I came to New York, probably around, I would say 2003, 2004, when I came back from DC, that's why I was focused on the academic side of things, because that's where the funding was going, and the startup culture, at least in New York, really didn't exist, or it was in its very, very early stages. I saw that, and I had my kind of epiphany moment. My eye openings happened right around 2008, if I was to kind of put a pin on it. And the firm I was working for was asked to work on one of the first and largest kind of New York-based commercial life science facilities. So I got involved in that particular project and was a part of the conversation and understanding a little bit differently how the science approached that. And you started to see some of the pharma saying, we're going to acquire smaller companies and move into some of these smaller venues to enable them to continue to go and to grow. And I think that's when it started to explain itself or show itself to me.
Yeah. In 2008, I was wondering which year this was. 2008 was literally the year that Lilly bought ImClone, right? So basically, you know, Erbitux and everything around ImClone was kind of that time. So you have kind of, you know, that demarcation of that was probably one of the bigger things in New York City biotech at the time. Regeneron, of course, was in Westchester and not kind of downtown or anything like that, but that was kind of probably pretty much a landmark event in terms of New York City biotech and the entire landscape.
And not long after that, I think it was Roche and Genentech had kind of come together as well.
Yeah. And then the Alexandria Center, the first tower went up, I believe, in 2010, or it was completed in 2010 and kind of leased out by 2011. So right around that time is, you know, really when some of the, I'll call them foundational things, because they were singular events, but they were big, right? And I think that kind of almost set the stage for, you know, you can do it and it can, you know, big things can happen here, right? And so it's interesting if we think about it from a real estate perspective, right? Like when Alexandria said they were going to build this tower, there's nothing else. There was no precedent. There was no, you know, there was nothing that said you can do it. And, you know, can you just kind of give us some perspective on kind of how big of a risk and how big of a swing that was?
At best I could tell you, I mean, huge, and honestly, not an easy thing to get our as a city, not an easy thing to get our heads around. You know, I think clearly there were some people with some foresight who said, let's do this thing. Let's find a way to make this happen. And, you know, credit to EDC as much as credit to Alexandria, both of whom are great organizations, for ultimately identifying that site and saying, okay, we're going to call this the East River Science Park, and we want to see this thing developed. And then Alexandria winning that bid and going forward with it and actually committing and doing that, absolutely, you know, hats off to them. It was a really, really bold move, to one that a lot of people, including, you know, some of the people on the teams were saying, this is great, but I'm still scratching my head a little bit about it. I don't quite understand quite what it is yet.
Right.
You know, and so.
Were there any, so Pfizer obviously was headquartered in the city, and ImClone, Lilly, but like there weren't any major buildings that were full of sort of independent, right, company labs. They were all inside academic centers at that time, right?
Pfizer's a great example, right? So the company I was talking about when I first started in Princeton, New Jersey, did a lot of work with Pfizer. For all I know, they still do. I don't, you know, but that said, all of it was in Connecticut, or a majority of it was in Connecticut, right? So they had an R&D facility in Groton, and then they built their headquarters were going to be moved, if I remember correctly, to New London, Connecticut, and then ultimately decided at the last minute not to do that. So there are two buildings on either side of the inlet there, one of which was supposed to be their headquarters, the other of which is their R&D headquarters. And so Pfizer was doing that, and I can remember one of the big arguments or conversations that surrounded building what they called the GDF, the Global Development Facility, was, well, you better make sure you put the helipad in there so people come back and forth from New York.
Right.
And that was the plan to some extent. It was, you can't do R&D and science in an urban environment because, as I was suggesting earlier, when you're dealing with chemistry-based science, that is a little bit more challenging, and it does have a higher hazard. And there were codes in place that said you can't build high-rise chemistry facilities. So it definitely pushed in that direction. So the idea of Alexandria coming in and saying, we're going to do a 16-story tower for life sciences, had a lot of people going, I don't know, I don't quite understand it. I could see it from a healthcare perspective where you say it's, you know, patient rooms and bed towers above, and then the labs are below. But it was a little bit more challenging to look at a mid-rise building, you know, a 16-story building that was designed for science. Not that they don't exist, but it was still a rarity, especially in New
Yeah.
York.
Did they
So
have, well, I'm
go ahead.
sorry, this is the danger of, letting the lawyer ask the questions about the size. So did it require like, and maybe this is more of an EDC sort of New York City question, but in order to put up buildings like Alexandria did, right? Whether it was Alexandria or future, right, buildings, did you have to have structural changes in, say, like building codes or safety regulations and all of that?
Well, at risk of kind of misstating what happened, I'm going to just give you my quick recollection.
Yeah.
There was a ULURP process. There was a rezoning that took place on that site. And there
Oh, I
was a
see.
ULURP process. And within that, or through that process, there were certain allowances that were granted to say, this is a little bit different from how you typically have done something in this region or this zone.
Right.
And it was done strategically. That was the whole premise behind the East River Science Park, was we're going to create that little enclave to create that center, that core.
Okay.
So to that end, yes. And New York City codes have always been New York City codes. They've always been different from, let's just say, everywhere else. So you'll hear, and I say it all the time, the difference between New York City and just about every other jurisdiction in the country is that every other jurisdiction in the country is IBC based, International Building Code. Whereas New York has always been New York City building codes. And even when we were doing the project and everybody was trying to figure out what do we have to do? We had consultants coming in from Chicago saying, okay, well, this is how we would have done it in Chicago. And then you had, because that's another high-rise environment. And you kind of scratch your head, go, well, why? Because this is building code in New York. So, I mean, that was probably my first introduction to some of these things, was finding a way to tooth all those codes together appropriately to say, this is justified, this is what makes this work, and ultimately making sure that we had all of the bells and whistles from a life safety and security perspective addressed first and foremost.
Yeah. We do it differently here. It's not Houston Street, it's Houston Street, right? So okay, so the first building goes up, and it's a little bit of, okay, we can actually do this here. So can you talk a little bit about what happens market-wise after that? I think from there, you probably have folks that now start to look around and say, okay, what else can we do here? Where's the ceiling? Like, how does the market figure that out, right? You now have one thing that proves you could put up a building and you can actually get some companies in with some labs. But, I imagine everybody else that wants to do it after that has to figure, well, how many companies are there and what's the actual appetite for this space? And with no precedent, how does the market figure that out? Because New York, you can't really go on. There's no real comp, right? It's not terribly easy to think about a different city and different dynamics and stuff, because the numbers around it hasn't existed before. Certainly not a city of this scale with this many people, so how does that happen?
It's still happening. I think when that gets introduced, when a project like that gets introduced, how does it change? It does change the way people think about, science, and they can, science being done in an urban environment, science being done at a commercial level.
Right.
And people can look at that building as an example of, hey, that's what I'm aspiring to, or that's a symbol of how this entire region can grow. And I think, a bastion, whatever you want to call it, but I think that that's something that was interesting, and did inspire some folks. And I think we saw, from a result of that, a lot more incubators really taking root. I don't want to say they didn't exist. I mean, SUNY has had incubators, for a long time. And so the idea that there was now a place for people to go to out of those incubators, that was something that I think changed the way the market looked at startup and commercial life science.
Yeah.
Just
although
I think you could argue
from a design standpoint, it's you can walk into SUNY Downstate and you can walk into J Labs, and those two things look very different, right? So there's incubators and there's incubators, and location counts. And this isn't to denigrate the lab space at Downstate. Actually, there's been good companies that come out of there. It's good space. You can get stuff done there. But there's a difference between kind of that and something that is a little bit more kind of, we're going to put this in the middle of Lower Manhattan to kind of catalyze the community. right? And those are two very, very different things.
I agree. And if I was to just kind of, and this is pure conjecture on my part, so bear with me here, but startup companies and incubators or otherwise who didn't see pathway, who didn't understand, where they could take their science, the choices were, all right, somebody either acquires my science and I stay here and continue to teach in the academic environment, which might be fine because I'm comfortable there. I've got tenure, whatever the case may be. Or I have to pick up and move. I have to go somewhere else. I have to go to Boston. I have to go to San Francisco. I have to go to, Los Angeles, San Diego. I'm not sure if I'm ready to do that. I like New York. I happen to be from New York. This is where I feel comfortable. So it actually, to some extent, not having that visibility or pathway to the growth, to the highway that gets you to that growth model, probably stunted some of the early stage academic and entrepreneurial life science in some of those early days. And the other thing to point to, I worked on, and it's funny because this was another beautiful melding of that academic and corporate world, but I worked on the Novartis Institute for Biomedical Research. And the incredible irony of that is just how intertwined, cyclical, and small our world can be. I worked on a project for, and I'm sorry to go backwards on us for a second here. I worked on a project for Shearing Plow in Summit, New Jersey. They had bought the SIBA campus in Summit, New Jersey. SIBA had merged with Sandoz to create Novartis. They moved to East Hanover, New Jersey, right? So my company was competing for both projects, and Novartis had said, we think there's a conflict of interest here for a number of reasons. You got to kind of pick which one you want. And we were already well embedded with the Shearing Plow folks. So we said, we're going to stick with that. Fast forward to where we are now, they had more or less taken all their R&D out of East Hanover and brought it to Cambridge. And now I'm being brought up to Cambridge as well to take part in the design process of their latest and greatest research facility. And in interviewing with the scientists at Novartis in Cambridge, half of them were saying to me, oh, I used to live in Clifton. Oh, I'm from New Jersey. Oh, they moved me up here. I'd love to get back. And even some of that was they didn't see pathway in this region to do anything about their aspirations for kind of being in this area. And I think that's something that this area has always kind of kicked themselves about. And I think that's exemplary of what we're kind of talking about a little bit.
Although, as a further aside, I may have been at Shearing Plow a shorter amount of time than you worked with them. I was there for 10 weeks before the merger with Merck was announced.
Okay. Yeah. So, and mergers is how this business rolls, right? I worked at a project at Kane University that was right next to the Shearing Plow Union site, and half the Union Shearing Plow folks came over and worked at Kane because that campus was closing. It was like, hey, I know you. And it was such an interesting dialogue back and forth, and Shearing got bought by Merck. Merck picked up and moved. Celgene came in and replaced them. Celgene is now BMS. I mean, it's just kind of the way things go, and so that's where I say it's like a little bit of a small world and a lot of people who have similar knowledge sets. And we all, to some extent, I'll say we all know each other, one way or another. And I think that that benefits all of us, but we would love to see that broaden. I think everybody wants to see that broaden and get new ideas to these things.
I think the other thing too, and I like the word pathway, right? Because one of the things that a big thing that fuels company creation and things is venture capital. And for a long time, the venture capital folks would do really the easiest thing in forming a company. The easiest thing was to put it in Boston or San Francisco or San Diego, because you could recycle a team that had done it before. The people were there, and you can make all of really kind of the easy excuses that, oh, it's easy to recruit, it's easy for this, it's easy because you could point to the scale and the ecosystem and say we're certain that we can do it here. And by the time we actually started to put incubators and lab space here, it kind of signified it was a viable option for them to stay here, right? Like if J&J believes that this is a good place for J Labs, well, gosh, that's a good destination for your startup. It's hard to argue against that, right? And that was really, in my mind, is a big way to kind of keep companies here, right? And now we've evolved to what I think is the enviable position of needing to kind of build things in the marketplace for them to go to when they graduate, or when they get bigger, or when they get successful. So it's a very enviable problem to have that probably nobody could really see coming in 2008.
Right.
Yeah.
Yeah, and it's funny too, because as an architect, a lot of the time, and as a developer, to be blunt, and say that there is no real, in the role that I'm in right now with Elevate Research Properties and Taconic, the reason I'm here is because that line is blurring between developer and architect in terms of what we should or could be providing. And I think that, some of the challenges, some of the benefits, I should say, maybe, was that line was blurring, and the line between incubator to pathway to growth actually exists a long time ago, and the institutions that exist. And when I say institutions now, I think I probably mean more private institution, but you've got conglomerate institutions. You've got things like Howard Hughes Medical Institute. You've got things like the Broad Institute. You've got things like, if you want to go back and speak to why this region has some importance historically, you got to go back to, for the sake of saying it, to the Bell Labs, which I know is not life sciences per se, but definitely was a cross-disciplinary or multidisciplinary incubator, for lack of a better way to say it, that took a lot of their IP and offered it out publicly. And people took that and said, I'm going to take their idea and run with it in this direction or that direction or another direction, which is one of the things that made, in my opinion, that collaborative environment so completely successful. Was their intent was, let's come up with ideas and then we'll figure out how to use those ideas, and we'll offer it to everybody else because maybe they've come up with something that we haven't come up with. And I think that, call it a corporate sharing that you probably won't see very frequently, is truly something that built this idea of cross-disciplinary practice. And it was between Bell Labs and MIT that were really doing a lot of that work. And the irony being that both of them were a little bit more engineering focused than they were science focused. And I think that aspect, or that viewpoint of it, enabled them to say, okay, this is not so much about science and research for the sake of science and research. This is about how can we make this a commodity or a product that can benefit people, and quite honestly benefit the corporation as well. I don't want to pretend otherwise, but I think there was something there. And I think that federal funding, which established the need during the wartime for research to be done, things like sonar creation, things like radar, things, encryption machines, and all these things that were federally funded through Bell Lab, or to Bell Labs and to MIT, ultimately resulted in an enormous amount of information that was then disseminated and creates things like, oh, well, we've created the internet from this. We've created the silicon microchip from this. We've got such incredible things that come out of that. So there is no one party that makes anything happen. It's cross-disciplinary within these organizations, but it's also federal funding. And, it is the support of an institution like an MIT that says, we're going to take this stuff and we're going to take it further. And when MIT got from the federal government the building they were doing all their experiments with for sonar, it became one of the most collaborative environments, cross-disciplinary collaborative environments the world has ever known. Building 20 on the MIT campus, which was a hand-me-down from the federal government. And from the architecture perspective, why this is so completely fascinating to me, the building was designed as a 3-year, 4-year building. It was designed not to last. Let's do it as cheap and ugly. We're going to tear it down anyway once the war's over, right? And the building wound up being handed over to MIT because they said, why tear it down? It's there. And MIT kept it up and running for probably another 30, 40 years before they ultimately tore it down.
Instead of a 3 to 4 year, it was a 30 to 40 year building.
Which is why architects completely panic when somebody says, oh, don't worry about it, it's temporary, because it's never temporary.
Never temporary. So that's why we see all the heavy concrete buildings out there.
And that's why we put so much darn money into those things. But the truth is, what I think is really instrumental to that whole conversation, whether it's Building 20 or the original Murray Hill Bell Labs, I shouldn't say original, but because they were in New York, but then when they moved to New Jersey originally, I think everybody has it in their head, that Eero Saarinen building that you see in Severance, that is so iconic for, architecture meets science.
Yeah.
But in reality, it was a building that was built, 15, 20 years earlier to that.
Mhm.
That was your traditional kind of brick, almost warehouse looking building, where they had these long, monotonous corridors by design.
Right.
And every scientist was required, not required as in written, it was an unwritten rule, an open door policy, literally leave your doors open so that anybody walking down that monotonous hallway has to pass all these open doors and peek into every one of those laboratories and say, hey, what are you doing in here? And it was that like intent to create this collaboration. In MIT building 20, MIT, to my understanding, just kind of handed space to people who wanted to do some sort of research. And they said, we don't know where to put you. We'll put you in building 20. And they just stuck everyone there, from mathematicians to musicians to artists to scientists. And when they needed more room, they literally took a sledgehammer and knocked the wall down. And then sometimes it was, whoops, I knocked my wall into my neighbor. Hey, by the way, what are you doing in here? And the craziest little things, like MIT building 20 had the first vending machine, is my understanding. And there was one of them in the entire building. And people would line up to get to the vending machine because they didn't want to leave for lunch. They didn't want to leave for lunch. And it became, the vending machine was the collaborative zone. You talk about people meeting at the water cooler. This was literally that. It was the vending machine. And so sometimes you just have to enable, and architecture just tries to enable that collaboration. The idea of an open door policy, engineers would be yelling at me, oh, I can't keep the airflows. I need negative pressure in the lab. I get that, it's not designed for that necessarily. But if we're thinking about how we can facilitate these things, then we can design around those things.
Well, I think this is probably a perfect segue to go to something that looks, I think, quite a bit better than Building 20 at MIT. Let's talk a little bit about West End Labs. Let's talk about that evolution. For edification of the audience, we're going to put the link to the website in the chat now. It's a facility over on 66th Street. I'm going to let Matt give the background, but having already taken a tour, it's spectacular. It's something where I'm genuinely excited for the kind of things that can come out of this building. So, and it's got a cool backstory. So why don't you give us a bit of the backstory on that?
I'll give you the back. And again, the joke was actually, when we toured Derek through the building, everybody told me not to talk because I talk too much. So I'm going to try and keep it brief. West End Labs. So Hudson Research Center. I'm going to go backwards for a second. This is my theme. Hudson Research was a facility that was an existing building that had existing occupants. We moved the New York stem cell into there. They came to us and said, this is one of the only buildings we found in New York City that we feel can actually suit the science that we've got. You meet the kind of the metrics or the parameters that we've done the analysis on. And so with that, we were able to do some, because it was an existing occupied building, we had to do laboratory spaces, piecemeal, a little bit here, a little bit there, because of the fact that the building was occupied with tenants. And so we did put $20+ million into infrastructure on the roof of that building. And it is one of the more energy efficient lab buildings that we're aware of. We won an award for it. Congratulations to JBB, our engineers, on that, for helping us with that. But air source heat pumps were introduced in that. That is a fully electrified laboratory building. And so, Local Law 97, which everybody kind of saw as this, problem, actually, in my opinion, is doing to some extent what it was really intended to do, which is to drive us in a different direction in terms of how we power buildings such as this. I think labs sometimes or were initially overlooked in that. And it was kind of ironic, in my opinion, that New York City is pushing for labs and then pushing for Local Law 97 without recognizing to some extent that the two may not meet. But that said, we did that for Hudson Research, and we said, boy, wouldn't it be a great opportunity for us if we could find a building that has similar robust, infrastructure potential, but doesn't have an occupant. So we could literally take a whole building, a whole building approach to it, and that was West End Laboratories. We took that building, I don't want to say we took it to its studs. We basically gutted the interior. We left the exterior up for the most part. It was built in 1929 for the Chrysler Corporation. It was, I always tell people, it was Detroit East. So if you imagine, right around that time, Chrysler's building the Chrysler Building. And they kind of said, okay, we've got this new product that has become available to the masses. Cars have come down in cost such that people are using them more now. We may kick ourselves for that later, but just the same, they said the market that exists for auto maintenance and repair was lacking, and people didn't find comfort at that point going to their kind of local mechanic. And Chrysler said, we want to keep you on brand. We want Chrysler owners to feel like they're being taken care of by Chrysler. So they built this facility, which was meant to repair cars, Detroit East.
Okay.
And as such, the building was designed so that cars could drive through it, including a corkscrew ramp at the back of the building to get cars up and down from floor to floor rather than employing an elevator to do that. We walked into the building and said, this creates, and at the time I was with Perkins and Will, who was the architect for both the HRC and this particular building, the West End Laboratories. We walked into the building and said, there's opportunities here that apart from the fact that this is now a vacant tenantless building, because ABC Disney had moved, they were in the process of moving to Hudson Square. So they vacated the building, but also, these kind of unique components like this corkscrew ramp are things that maybe we can kind of lean into and own the history of this building in this neighborhood. We didn't want to tear the building down. We wanted to preserve the building, one, because it made better sense for us financially at that point, but two, there was no real reason. It hit all the metrics. It had the great floor loading capacity. It had the good vibration performance. It had great floor to floor heights. There was no reason to tear it down. And it was a part of a neighborhood that we didn't necessarily want to completely change by putting some foreign UFO object into it. So what we said was, let's take the building, maybe we reclad the exterior, and what we did was an overclad. So we left large portions of the exterior in place. We did tear down the windows, which were designed to make sure cars didn't roll off the building and out into the, so they had big high windows, so 48-inch.
That's a thing people try to avoid. They try to not do that.
Cars.
It was harder to do apparently in 1929. Today, our parking garages were able to do that pretty, no, I'm kidding. But so the windowsills were up at like 48 inches. So we said, well, that's not a good atmosphere, right? We need to do something about that. So we tore that down. We took the sills down essentially to the slabs and replaced that with more glazing, more transparency, more natural light access. But other than that, the building, the other major move we did to the structural element of the building was we took all of the core elements, the egress elements that were on the perimeter of the building, we pulled them into the core so that we could open up more of the perimeter. We reclad the building, so it's a completely new clad, energy efficient building. And then we put infrastructure in place, like on the roof and then vertically down serving the rest of the building. That was lab infrastructure. So we have all the air handlers there. We've got the fume hood exhaust fans there. We've got all of the chiller boiler requirements there for laboratory space. We planned that based on some of the things that we had learned at Hudson Research Center with regard to the lab office relationships and ratios, with regard to biology versus chemistry. And we said, okay, we understand enough that we're going to take a shot at the infrastructure requirements of a laboratory building such as this, put that in place. But we wanted to keep some of the elements, the atmosphere, the energy of the building that existed from 1929. So we kept that corkscrew ramp. Which for me anyway, and I mentioned Novartis before, they had a building, for example, directly across the street from where my project was, called the Necco Wafer Factory. And they put a hole right in the middle of the building, created an atrium, and played with that, architecturally as this kind of candy element. And I said, that type of space is something that maybe we can replicate here in New York. New York needs that type of space. So that was kind of my pitch anyway. There was a drawn out conversation. I don't want to pretend otherwise, but my pitch for keeping that ramp, and everybody kind of said, well, how are you going to make that functional? How are you going to actually take that space, this corkscrew ramp, and instead of making it a museum piece, making it a functional piece? So we introduced it as what we call the helix. No puns intended. Well, all puns intended.
Yes, all. I was like, no. You got to own that, Matt.
But what we've done with that space in our pre-built floor, which is 54,000 square feet of pre-built laboratory environment, was we created a collaborative zone. So the center of that corkscrew is an open meeting environment, a forum type of an environment. And then up and down around this kind of ramp, we've placed pods or conference rooms, enclosed conference rooms, so that individuals on that floor can go and either book and use a conference room as a private meeting, or use that open forum environment for collaborative purposes, whether they just want to go out there and have their lunch, or they want to have an impromptu, all hands touchdown meeting or something to that. This space, which was otherwise doomed to either being torn out or underutilized, we think we found a way to make that a space that can be utilized, becomes a focal point for a floor and for the building, and also speaks to its history. So we really kind of like that aspect of it. And we leaned in on that a little bit. This is a building that has what I would call a gracious lobby. We have what we call Connect Well, which is about a 15,000 square foot environment for meetings. It is a conferencing center. Essentially, we've got a 300 person capacity down there. We just recently had an event with BizNow at that space, and it was kind of our trial run, if you will. Yeah. And I think it proved successful, and we were pretty happy with that. So we've got different functions to that. And the other thing we were able to do, because we were able to think about this as a vacant building, knowing all the parameters that we wanted to put into the infrastructure, was make sure we saved a chunk of roof space. So we have what we call Parkwell, which is a rooftop, outdoor roof deck environment, or a roof terrace. That was kind of the corner of the building, the 10 to 15,000 square feet corner of the building that we were able to preserve with views of the Hudson River. The idea of having that area of respite, that get outside space. One could argue, oh, well, COVID, amps up the need for things like that. But the truth of the matter is that was a need that has always existed. And I've never not seen outdoor space get, underutilized, especially in a place like New York City. So it was something we were thinking about in 2019 before COVID had hit. So, yeah, it's a great little piece and a great little way to kind of take a break from your desk, which we think is incredibly valuable.
We spend so much time in offices, but perhaps less time than we used to, but that you're so connected to it. I think one of the interesting things is when you create a space that people want to go to or like to be in when they actually go to work. There's a lot of that where it's not a soulless, monotonous, infinite hallway. It's something where you have spaces to your conference rooms are different, your sitting space is different, the roof space is different, and it's something where you can actually be there without thinking that you're in a drab box that you can't wait to get out of, and you've nailed it, right? And I also love the event space as well, because there are there's a ton of events in New York. Every one of them needs a venue, and in a lot of way, for a long time, biotech events, it was at some hotel, right, which is fine, right. But I think as an industry, we get a lot more of a richer experience when the things that we do as an industry are around other things in the industry, right?
Yep, we see it as an anchor point for the community, not just the West Side cluster that happens to exist there. I mentioned HRC before. HRC and Well are 10 blocks apart. So it's becoming part of a cluster. You got Mount Sinai West up there. You've got a number of different opportunities that are up there as a cluster. And so we saw this as an anchor point for that cluster. This is a 400,000 square foot building that has substantial heft that can really support what we were talking about before, that pathway to growth, that the growing of a life science community within a cluster or within a building specifically.
Yeah, sneaky good location too.
Yeah, I was going to ask about that.
Sneaky good, right?
Yeah,
right on the one train. And I didn't appreciate when I got there that the 66th Street bus goes straight from Cornell, Rockefeller, etc., and lands on the doorstep in about 10 minutes.
Yeah, the 65th Street transverse connects east side to west side in under 15 minutes. It's really advantageous.
Yeah. And then and then so I know we only have 5 minutes, but so as far as a sort of takeaway, what did you all do differently in West End Labs based on the knowledge you've gained both from Hudson Research Center, from other projects that have gone up in New York? What did you do differently for this project that maybe we didn't know about sort of as the earlier lab space and office space for life sciences grew in New York?
Well, I think rather than say what we did differently, I would say what we did consistently is some of those lessons learned. So flexibility is a key element to this. And I know there's a question out there about that.
Yeah.
The flexibility aspect of this is as a commercial developer, life science developer, that is something we need to be very, very mindful of, because, the tenancy has the potential to be so completely varied. The science that the tenants will undertake has the potential to be so completely varied. We have what we call, or I've dubbed it the 80-20 rule, which is to say, I'm going to get you 80% of the way there with our pre-built environments. And the pre-builts are absolutely crucial, in my opinion, to helping kind of seed the clouds of this market, because one of the big reasons a lot of organizations have left the environment, the city, is because of the absolute lack of genuine lab space. Outside of academic institutions, there really wasn't any in New York. So we felt it was important for these pre-built environments because of speed to market. Because when a company gets a Series A, they need to move. Or when a company has said, "Hey, your time in the incubator is up. You've been successful. Congratulations, your time in the incubator is up." They need to move.
Check it out.
Right? And so we needed to make sure that we had space available for them to move into. And the 80-20 rule on that is, as we've designed, as flexible as we can imagine to be. But we never say we're going to get you 100% of the way there. This lab isn't the lab that's going to suit you perfectly. It's 80% of the way there. You're going to come in and you're going to have to maybe manipulate it up to 20%. And these are just, pulled out of the air numbers. But the idea very simply is, okay, it's not, you could go in, you could start your science, you could start setting up your equipment. But the reality of it is, we're not necessarily saying we're going to thread that needle perfectly, and that's something that is important. But rather than waiting 18 months, given all the lead times that we're facing nowadays, you could be up and running in 3 months, 4 months. It's an enormous difference. And the space being available as a pre-built to move in and get comfortable with it while you're doing that enables you to get out of your existing space quickly. And I think that's also important. I think you mentioned, what do we do differently? I think one of the things that, as we're kind of running out of time here, I'm just going to jump to it real quick. One of the things we're thinking about doing differently, in fact, is we've got spaces within West End laboratories that have flexibility to take on different functions, specialty laboratory environments. We've got high bay spaces. We've got a second floor that has the capability of a direct drive-in laboratory environment. So if chain of custody is a concern for you, for example, you can drive directly into a second floor and have a diagnostic laboratory in there if you needed to or wanted to. And clinical lab space and clinics and clinical trials, New York is king by a long shot in clinical trials. Is where the translational stuff comes back into play. And I feel like that's one great opportunity of the many that exist for New York City to kind of take its place in life sciences, is the abundance of healthcare organizations, I'm looking out the window here, the array of people that exists that will enrich a clinical trial. New York can and will be and is the kind of clinical trial capital. And I think that translational research element, when you start to introduce things like blood testing, AI, as it basically feeds into drug discovery, there is a real opportunity for New York to kind of embrace that and take that a step further. Our project at Iron Horse Labs, which is on the east side now on 94th Street, 309 East 94th Street, we've designed in, similar to what we did at Well, but to the next level, the potential or the opportunity for high bay space, like a 30-foot clear space, where we understand you could get some high resolution imaging, like a cryo-EM, if that was something that was crucial or needed, or you could position that for an animal research facility, or you could position that for, maybe more so and more importantly, a GMP facility. And I don't mean to say we're making manufacturing and producing things in the scale that the global industry needs and is asking for, more so in that scale up translational R&D, proof of concept sort of stage, clinical trial batches. So I think that's something that we're introducing now with Iron Horse Labs as a different way to look about these things.
Yeah.
Here, you want to wrap us?
Yeah, I can wrap up. I mean, This has been fantastic. We usually kind of save time at the end, but you've covered all sorts of things. And I think the big takeaway here is that, nobody could have seen this in 2008, right? We've got things that are just getting off the ground now. And I think you guys have created some places that I think are going to do exciting science, and I'm excited to see it. So thank you so much for joining us. There is no humidity in New York today. It's a gorgeous day. So I hope everybody goes out and enjoys one of the nicest last days of summer.
So
Thank you
apparently
all. If you want to take a look, feel free to reach out. We're happy to give looks.
Yeah, we can do that. The humidity is still in DC where I am. So I will try to keep it down here.
It is your fault for going to DC in August. Have a great day, everybody.
Have a good day, everybody.